Welcome to the Backyard Bounty Podcast from HeritageAcresMarket.com, where we aim to educate and inspire you by sharing practical information to help your homestead thrive. And now, here's your host, Nicole.
Hello, everyone, and thank you so much for joining me for another episode of Backyard Bounty. I'm your host, Nicole, and today I'm joined by Alison McAfee. Today we are going to talk about queen bees. I'm really excited to have Alison with us on the show today. She recently did an amazing talk on NPR, which is how we learned about her, and she's incredible and very well experienced in honeybee research. So excited to learn from her today. And Alison actually is currently studying hygienic behavior and queen health and reproduction. She also currently writes for the American Bee Journal and has some pieces in the Scientific American. So with all of that being said, Alison, thank you so much for joining me today.
Yeah, thank you for having me.
Of course. It's great to have you. I'm excited to talk to you. You know, as much as people love bees in general, I feel like people like the queen bee the most. And so lots of questions I've come to find along the way. People always want to know about the queen. But before we get into that, can you give us a little bit more background on what exactly you were studying and some of the things that you learned along the way with your honeybee research?
For sure. Yeah. So I actually started out by studying hygienic behavior. That was for my PhD research, which has been given more attention recently than it ever actually was during my PhD, because all of a sudden we're very interested to learn about how insect societies deal with disease outbreaks, given what is going on in human societies currently. But I have since transitioned to studying queen health. And I'm studying mainly factors that affect her reproductive quality. So like things that can interfere with her fertility. Queen health has been anecdotally declining. So something that I hear a lot from beekeepers is that queens just don't last as long as they used to. So my research is really about trying to figure out why that is, what's happening to the queens to kind of cause their early decline.
And so what are some things that you've found so far?
Yeah, so first I started studying temperature stress because that is maybe a surprising way that queens can become stressed. If you've been in a bee colony, or not yourself, but if you've been sort of playing around with a bee colony, then you know that inside that colony, the workers do a really good job of regulating the temperature normally. So temperature stress might not be something that you think of as something that the queen could be exposed to, but we humans really like to stuff queens in tiny little cages and then ship them halfway around the world. There's an international queen distribution industry that some countries, such as Canada, where I am, really depend on. So Canada imports around 250,000 queens every year from international sources, usually the US, but also as far away as Australia and New Zealand. So when queens are in these tiny little cages, they don't have that worker mass around them to really thermoregulate. And they're basically at the mercy of what the ambient temperature is in the plane or in the warehouse or in the van. So we've found that extreme temperatures happen in queen shipments somewhere around 10% of the time, which I think it's enough to be worried about anyway. And the other reason why I started with temperature stress is because we know from work done in all kinds of different animals, from fish to birds to mammals, that heat stress can kill sperm cells. And that's particularly important for queen honeybees because, as you might know, queens mate during one short period early in life, and they acquire all the sperm that they need for their entire lifetime supply of eggs that they'll ever lay. And they store it in the spermatheca, this specialized storage organ in their abdomen. So if the queen is exposed to something that kills sperm cells, that means that those sperm she's stored inside her could start to die. And if that happens, she can't go and mate again to acquire more. She only has that one chance to get all the sperm that she needs. So if something kills the sperm she's stored, then that's really bad news for her ability to maintain a colony, keep being able to lay enough eggs.
I know that the queen has an ability to lay fertilized and unfertilized, but I'm assuming she probably doesn't have any way of knowing whether that sperm's viable or not. She just lays the fertilized egg without knowing one way or the other.
Yeah, that's a really good question. So the way that it works when she lays an egg, you're right, she can like decide whether she's going to try to fertilize an egg or not. And the actual mechanism of what happens is like when the egg is physically traveling down the oviduct, it'll hit this kind of fold in the tube and press it against this little sperm pump. And that's a little tiny muscular pump that can force fluid from the spermatheca, where the sperm are stored, into the oviduct. And so that is how she can fertilize the egg, is by releasing this set amount of fluid. So because she's just releasing like a volume, you're right, she can't control whether that volume contains a viable sperm or non-viable sperm. So some really cool research done by Boris Baer, who is in California, showed that on average, a well-mated queen will release about 2 sperm. So in that volume, there's usually about 2 sperm cells. And if everybody's alive, then it's kind of like 1 sperm cell is to fertilize the egg and then the other one is for good luck. But as you can imagine, once the sperm start to either die or run out, just as she gets older and uses them up, then chances become higher and higher that she'll lay an unfertilized egg by accident.
Okay, interesting. So then at some point in time, the workers just realize that she's laying unfertilized eggs and work on superseding her?
Yeah, that's kind of one of the going ideas for how workers might be able to recognize that a queen is declining, is by this increased frequency of laying unfertilized eggs. The way, this wasn't my work, this was done before I started studying this topic, but the way the authors described it was that this could be like an honest signal of fertility decline that's perceptible by the workers.
Sure. Interesting. And I had read about the temperature. I'm not sure where. It might have been in my master beekeeping program with Cornell, but I thought that was really interesting because so many people do rely on packages, like you said. And I mean, everybody, I think, would probably assume pesticides or something like that. But I don't know that temperature really is what anybody would think of. So how did you even come up with the possibility of temperatures with queen bees? Just, I mean, I know that you mentioned earlier that, you know, it's known in other species, but is it just something that you noticed that they were getting overheated in transport, or how did you even think of it?
Yeah, that's a really good question too. So this actually originated by work that one of my advisors started doing. So again, before I was brought onto the project, it seems like everything in science is like a legacy project. You know, it's never just one person like doing something by themselves. I'm sort of following in the footsteps of a lot of people who have really laid some good foundations here. But Jeff Pettis, who used to work for the USDA, he published a paper in 2016 where he was looking at what the differences were between queens that were rated as failed and healthy by beekeepers in different locations. And he found that consistently the failed queens had low sperm viability. So that kind of clued him into sperm viability maybe being one of the things that was causing the queens to fail. And then the next question is, okay, what can cause sperm to die? So observation that this temperature stress can kill sperm in other animals made it a natural thing to look at. And something that both he and I, and a few other people by now actually have done, is include temperature loggers in actual queen shipments to see what the temperature variation is that actually happens in just regular, like the way that you would normally ship a queen in the same kind of package with the same couriers or whatever, and just see what they get exposed to. And almost every time we do that, at least one of the packages will end up getting a temperature spike. So the sort of smoking gun, though, was when he sent queens off. He had 2 packages of queens that he sent off, one by UPS and one by USPS, and had temperature loggers in both of them. These were queens from the same place, from like the same batch, so they should have very similar sperm viability in the beginning. And then he sent them off, and one of them actually happened to have an extreme temperature event happen during the shipping, and the other one didn't. And then when they looked at the sperm viability of the queens in those 2 packages, they found that the one that had the temperature spike had lower sperm viability. So then it's like, okay, it's all coming together. This is a real risk, and it could potentially really like have an impact for the end user, the people who receive the queens too.
How interesting. So is that kind of the majority of your work right now, or are you looking at other causes as well?
Yeah, so I have looked at pesticide stress as well. You mentioned that as being like one of the things that people might immediately kind of think of. And that's for good reason, because there's a lot of data out there showing that pesticide exposure is linked to queen events or declines in queen quality in a number of different studies. And so I was interested to see if pesticide stress in kind of a realistic dose could actually also kill sperm cells, because we know there's only really one pesticide that has yet actually been shown to kill sperm. And it's not one of the ones that is most often found inside beehives where a queen may actually come in contact with it. So I looked at the effect of a mixture of the 9 most abundant agrochemicals, which includes everything from fungicides, herbicides, insecticides, miticides, and in some cases, they're degradation products. I looked for an effect of if I essentially like dabbed this mixture onto a queen, does that do anything to her sperm? That would be kind of like a worst case scenario. And I haven't found any effect whatsoever of that. So what that tells me is that this really strong relationship that we know exists between pesticide abundance and queen failure is probably due to indirect impacts on the workers. So pesticide stress, like workers are the ones that are most exposed to pesticides because they contact it in their environment, and they're the ones who are like chewing the wax and preparing the bee bread and all that kind of stuff, whereas the queens just fed the workers' glandular secretions. So the workers you'd really expect them to be the most impacted. And it looks like what is happening is probably the pesticides are causing the workers to either produce different secretions that they feed the queen. So maybe it's like not as nutritious or something like that. Or it could be interfering with their ability to perceive the queen and like give her adequate care that way. But whatever it is, it doesn't seem to be a direct effect on the queen herself.
Wow. Yes, that's a good thing, but how interesting.
Yeah.
My mind is kind of swimming with, with questions. Like I kind of had a topic kind of that I wanted to stick with, and I could very easily ask so many other questions, but that's interesting.
That's my constant state.
Is there anything that you found along the way in your research that was maybe surprising or more unexpected than not?
So something that was both surprising and disappointing, I guess, was that one of my projects is to try to come up with a panel of molecular markers for different forms of queen stress, essentially that I can measure in the lab, and then which indicate if a queen had been exposed to a particular stressor or not. So the idea is that if a queen sputters out and she's no good anymore to a beekeeper, instead of squishing her and replacing her with a new queen, like you would need to do if you're running an operation and need to keep the hive going, they could send that queen to me, and then I can run these tests and try to tell them what might have happened to her. So that's one of my ongoing projects. And I started out by temperature stressing the queen. So either exposing them to temperatures that were too hot, that were too cold, or a pesticide exposure. and then comparing those to queens that weren't exposed to any stress to see what proteins become like more abundant or more activated in the queen that may indicate, that are unique to each stress. Mm-hmm. And so I was pleased with the initial results because I heat shocked the queens and then saw some specific and predictable proteins that became very highly elevated. So I thought, this is great. This can be maybe a good indicator for heat stress in queens from the field. And then I looked at queens that I sampled from throughout BC, and this was a lot of queens. This was like over 50 failed queens, and I also had over 50 healthy queens. And I looked at these two markers that I was hoping would work, and was really surprised when I found that over half of the failed queens had elevated levels of these two heat stress markers. And at first I was like, that's crazy. How could all these queens be heat stressed? It just doesn't make sense, because I also had temperature loggers in the shipments that sent them to the lab, and I knew they didn't get too hot during transit there. So it was just very puzzling. And then it wasn't until another student in the lab who actually was interested in looking at viral abundances in the queens, she did some analyses to check if the queens had virus infections. And they did. The failed queens were definitely sicker. They had higher levels of viruses. And actually, these two markers for heat stress are also elevated when the queens are sick. So essentially, this whole concept of being able to look for markers for particular stressors is much more difficult than I initially thought it would be, because there's actually some problematic overlap between different stressors for the queens. So it's complicated things a lot. It's both really interesting, because now I'm getting all these crazy ideas of, well, maybe if I heat stress a virgin queen, then that can help protect her against viruses that she may acquire in the future, things like that. But that was very surprising. I didn't expect that.
Yeah. Do you find that many queens have viruses? Do you look at that side of things as well?
Yeah. So since that test, I have become very interested in viruses now. I haven't yet found a queen that actually is symptomatic of virus infection, but we've tested, I guess we've probably tested around 150 queens now. And a lot of them do have infections. It tends to be mostly deformed wing virus, as you might expect, because deformed wing virus is everywhere. But they also carry sac-brood virus and black queen cell virus. Even as adults, they can carry black queen cell virus, which was pretty surprising to me. Now, with this knowledge that these queens have all kinds of sublethal virus infections, now I'm wondering what kind of impact those infections are having on their ability to reproduce. And we've found some really interesting relationships where queens that are more infected with viruses tend to have smaller ovaries. And as you can imagine, that's kind of now impacting the other side of reproduction, which is egg laying. So we were looking at sperm viability at first, and now the path has kind of led us to look at impacts on egg laying too.
So with the viruses, are those transmitted mostly through varroa or something where they're getting them from the workers, or do you know how those are being transmitted?
We don't have a way of sort of repainting the transmission route for those particular queens, because we just sampled them when we had the opportunity to sample them. But some work has been done on potential virus transmission routes, just in general. And one of the ways that queens can get infected is through mating. So viruses can actually be considered sexually transmitted diseases for queens. I am not very familiar with other routes, but surely there are other routes of infection. Just this sexual transmission is something that's obviously unique to queens and is pretty intriguing for that reason.
Yeah, that's not the route I would have assumed at all. I think what you're doing is really intriguing, and how neat.
Yeah, another neat thing is that, so I do a lot of this technique called proteomics, which is a lot of people are familiar with genomics now, which is like studying all the genes in an organism. Proteomics is like studying all the proteins in your sample. So I will often do things where I like take a tissue sample, say the spermathecal fluid, and then look at all the proteins that are there and what their abundances are, and then try to link that to something else to do with the queen, such as the sperm viability, maybe. And when I just look at what proteins are present in the spermathecal fluid, there are actually a lot of virus proteins in there. So that is like further evidence that viruses can be transmitted sexually, because how the heck could virus polyprotein get inside the spermatheca?
Right. Wow. So obviously, we've talked a lot about the things that can go wrong with queens, but to maybe kind of look at a different angle, can you kind of elaborate on what the queen's role is in the colony and why these are so important? I mean, obviously, laying fertile eggs is important for the colony, but just kind of a little bit more on the queen's role.
The name queen is kind of almost a misnomer, because to be queen is actually really to be a slave. You're a slave to laying eggs all day, every day, as many as you can. And there's virtually no time to do anything else. So the workers feed the queen, they groom the queen, they will like carry her poop away when she has to poop. They do everything for her so that she can just focus on laying eggs. She's the only individual in a colony normally that is producing eggs. So when something happens that causes her fertility to decline, then as you can imagine, the colony population starts to dwindle, get smaller and smaller, and less productive, which is both a problem for beekeepers who need these bustling, productive colonies to make enough splits to put them into pollination, or to have a big enough colony that can gather honey that you're going to sell for your income. So when the colony population dwindles, even if you can rescue it later on by, say, requeening, it's still a pretty big problem for your bottom line as a beekeeper.
Sure. And I imagine that these issues with the queens, though, not so much the temperature, but they're not isolated to managed colonies. They assume that even the feral ones and, backyard beekeepers and everyone is impacted, obviously.
Yeah, you know what? That's such an interesting thought. We have very little idea of what's going on with the queens and feral colonies, because you just can't study them. Like, you can't get into a feral colony most of the time, right? It's like in a wall of a house, or it's inside of a tree or something, and you can't actually really study what's happening to those queens. But that is a really interesting question.
Well, maybe we'll learn more someday. Obviously fertility is super important, since that's her main role. So that makes sense as to why her fertility is so important. But without the queen, you're not going to have a colony. And I don't recall if you said this in the beginning. Do you know about how different their lifespan is now compared to, I don't know, 5 or 10 years ago, or however long?
Yeah, this is the kind of thing that I really wish that we had solid data for, because unfortunately, 10 years ago, people weren't thinking that this would be a problem in the future. And so there isn't really good documentation on how long queens used to live. But what we know now is that, well, I guess as far back as 2013 is when this paper was published. Dennis Van Engelsdorp did a survey of beekeepers in the US. And in that data, one of the findings was that 50% of the time, queens are replaced when they're 6 months old or younger, which is very, and I must qualify this with that this was data for those particular operations, and I don't know exactly how representative those are of the nation. But regardless, even if he didn't choose the operations that have the worst problems, so it's somewhat representative at least. And that's a pretty worrying stat, I think, because queens have the potential to live. The oldest reported queen was 8 years old, which is crazy. That almost never happens. but they do have the potential to live like 5 years. So if half of them are being replaced when they're 6 months old or younger, then that's like, it's suggesting they're really not like living up to their potential.
Sure. I guess I just assumed that, like in the commercial setting, it was just once a year, every spring or whenever they chose to requeen. I didn't realize that it was that frequent.
Yeah, no, it's kind of worrying. The other thing is that makes it sort of hard for me to answer your original question is because I think in commercial operations, they often do kind of like a mass requeening where just like everybody gets requeened on a set schedule because you don't have the time to go through each colony and see what each one needs. So for that reason, it's also like we might have an idea of approximate requeening rates, but it's really hard to tell if it was actually like necessary for those colonies.
Sure. Is your research mostly done on queens that were artificially inseminated, or do you look at queens that mated naturally?
I've actually never worked on artificially inseminated queens. I would really like to because there are all kinds of neat experiments that I want to do. I first need to learn how to do it. But yeah, to answer your question, all the queens that I've studied so far have mated in a natural setting.
So when you look at these queens, are they coming from like test yards so you know more or less the drones that they're mating with, or are they just kind of random drones from wherever?
Yeah, so that is also a really good question. I have not yet played with controlling the drone source, although it is a direction that I am thinking of taking the research in the future because I had some really strange results this past year where I experimentally heat stressed a bunch of queens and then didn't observe the characteristic decline in sperm viability that I was expecting to, which is weird because we've seen this over and over and over again in other queens. And so it got me thinking what could potentially be different about these queens compared to other ones. And something that we've never bothered controlling for is exactly what sperm these queens have stored inside them. And it's totally plausible that not all sperm is made equal. There's large variation in drone quality of the drones themselves. And there's also large variation in the viability of the drone's sperm. So it wouldn't be crazy to think that there's variation in the tolerance of the sperm cells themselves to different temperatures. So something that I would like to do is to actually do some artificial insemination, controlling drone sources, and then seeing if the sperm viability drop off in queens with those different sources is the same, or if it depends on where the sperm actually came from.
Do you work, or have you looked at a variety of different honeybee races or species or whatever you would like to call them, or have you just kind of focused on one variety?
I have done heat stress experiments on queens from a variety of producers. So I haven't done genetic testing to like confirm exactly what genetic makeup they were, but I did, for example, heat stress locally produced queens, and I've heat stressed queens from California and ones from Hawaii. And with the exception of this weird group I had last summer, across all of them, we see the same stimulation of the same proteins and a very similar drop in sperm viability. So that does seem to be like something that affects almost everybody, though apparently not this weird group that I had. We still haven't figured out why it didn't have the expected effect for those particular queens.
Always keeping you on your toes, I guess. It's always going to be something unique. I don't know. Wow, see, I almost did it again.
I leave you speechless.
You do? No, you do, because it's so fascinating, and it makes me want to ask so many questions, and what you're doing is just so neat. I think that just the things that you learn and the unknowns and the questions, and I've run out of the ability to make sensible statements, I guess.
Yeah, there's an endless number of things to follow up on researching.
So what are some things, I know that you've touched on a few of them, but what are some of your goals moving forward and some questions that you'd like to answer and just kind of, your general plan?
Yeah, man. I mean, the current state of the world is always changing my general plans going forward. um, the university's shut down, then it's not, and then you're not supposed to go to work, and then you can go to work. So things, as you can understand, have been a little bit up in the air lately. But something that I would really like to do, or at least start working on this summer, is to follow up on this idea that I might be able to protect virgin queens from virus infection before they go off and mate. Yeah, I'm going to start looking into that this summer by, for example, heat stressing the virgins when they're, early in life, maybe let's say 4 days old or something like that. So when they're young and they haven't yet gone out to mate, and then infecting them with a virus in the lab, but you can like inject them with virus or expose them in some less invasive way as well. So then I want to see if the heat stressed queens end up doing better than non-heat stressed queens when they need to fight off that infection. So I haven't started working on this yet. There are so many kinks to work out really before the experiments fully get underway. But that's sort of the direction that I'm looking at taking this. And this other project of trying to come up with a diagnostic tool to figure out different causes of queen failure is a little bit at a standstill because of this problem with overlapping markers for the viruses and the heat stress. So I still really want to be able to provide such a tool, though, because I think that would not only be cool scientifically to be able to like actually figure out how many queens are failing because of stressor X, Y, or Z, but it would also be really, I think, useful and interesting for beekeepers too. So I'm trying to figure out a way forward with that. It just might take some much more complicated statistics than I originally thought.
Sure. If you could figure that out, I think that'd be an amazing tool, not only for, again, backyard beekeepers, but the people on a larger scale, if they can figure out, well, this is what's wrong, then this is how we can fix it. But you have to be able to figure out what's wrong to be able to fix it. The most.
Yeah, exactly. And temperature stress, I think, has been a really like shrouded problem. Like we just haven't really thought too hard about that being a problem because it's not obvious. Like a queen that has been heat shocked or cold shocked looks exactly the same as a queen that hasn't. So there's no way visually to tell that something has happened. And then, of course, if she ends up not performing very well, as anybody who has had any experience beekeeping knows, like there are a whole slew of potential things that could have gone wrong. And you usually have very little capacity to actually narrow it down. So, I think that this temperature stress thing has probably been impacting our industry for a long time, but it's just that it's been virtually impossible to diagnose.
Do you think that it would be something where just like a larger queen cage, or where they would need to maybe ship them in a larger container? Do you think it's something that could be relatively easily mitigated?
Yeah, actually, I do. So at least in terms of cold stress, that would be pretty easy to protect them against. Some researchers in Quebec have done some experiments where they shipped queens in different types of containers, and they got the best results in terms of like the temperature remaining the most stable, even if they threw them in basically a refrigerator, when they added a few hundred nurse bees into the box that the queens were kept in, like just loose nurses. That is a potential option for domestic shipments. But as far as I know, for international shipments, the queens need to be like inspectable by whatever agency is in charge of the import inspections. So having loose nurses is problematic for that. But at least in terms of domestic shipments, that's one like pretty easy way. I think that the risk of cold stress could be mitigated. And for heat stress, I think that people are pretty innovative, and I'm sure that there's some kind of material we can use that would both be friendly to the bees and the environment and protect against external heat. I haven't tried to figure that out yet, and I don't know anybody who yet has, but it seems feasible for sure.
Interesting. Yeah, I guess prior to talking to you, I didn't know that they shipped bees internationally, so that definitely adds to the challenge, I think.
Yeah, so in the US, the vast majority of queens are supplied by just a few like big producers. I think there are definitely the ones in Hawaii and California, which are the ones that I deal with most often. But then there's also Florida, Georgia. I imagine that they, probably being in the warmer region, probably will produce queens for the north. But yeah, it may not be international movement, but there's still like a large dependence on like a few producers dispersing them to nationwide.
So obviously there's a lot going on and a lot to learn as well. What's the best way for people to follow your work and to see what you come up with in the future?
Well, you could follow me on Twitter. I am fairly active on Twitter, whether I like it or not. My Twitter handle is just Ali_McAfee. I also have a blog, AlisonMcAfeeBlogs.wordpress.com, where I post some of my recent articles from American Bee Journal or elsewhere if I happen to have published something that isn't protected by copyright. That's probably the easiest way. I would like to add just one more thing, though, about the queen shipping. If you are a backyard beekeeper and you're sort of like doing this for fun and you don't necessarily depend on getting a huge number of queens at a specific time of year, then I would highly recommend trying to get a local queen, because then you can just avoid the problem of shipping altogether, as well as support more local businesses. That is definitely something that I would recommend.
No, that's really great advice. I think that most people just assume right off the bat that they need to buy a queen or whatever. And I think in a lot of backyard beekeeper situations, they can have really good luck raising their own. But I also really like the idea of supporting your local beekeeper too, because we all know that we're all poor beekeepers. There's not many rich beekeepers. So definitely like your advice of supporting local producers.
Mm-hmm. And just to add one more thing to that, even if you get a queen from your local bee supply store or outlet, do still check to see where it came from, because sometimes these stores will import them or bring them in domestically from somewhere far away. So best would be to go like directly to a queen breeder that you know and trust, or check to see that your local supply store is actually selling the locally produced queens.
And I would assume that local and state beekeeping clubs could probably put people into touch with queen breeders. Do you know of any other resources to find if you have local suppliers?
Yeah, that's probably the best one. Bee clubs are so helpful to beekeepers at all levels, and there's so much knowledge carried and passed on through them. So yeah, I agree that's probably the best place.
Well, Alison, thank you so much. In the show notes, I'll put your Twitter handle and your website so that people can find you there. And I'm excited to see what you come up with in the future in your research.
Thank you.
Yeah, thank you so much for joining me. I really appreciate your time.
You too. Have a good evening.
Thanks, you too. And for those of you joining us, thank you so much for joining us for another episode of Backyard Bounty, and we'll see you again next week.
For a behind-the-scenes sneak peek, receive episode notifications, or leave a question or comment for the show, text the word podcast to 719-292-3207, or visit heritageacresmarket.com/podcast.